Today I'm going to talk about transcription in prokaryotes. Transcription is the synthesis of RNA via a DNA template in the cytoplasm of a prokaryotic cell. There are 3 steps to transcription. The first step of transcription is initiation, where the RNA polymerase finds the promoter region of the DNA. The second step is elongation, where the RNA polymerase makes the RNA strand complementary to the template strand of the DNA. Lastly is termination, where Rho attaches to the RNA or hairpins are formed to stop the RNA synthesis. Here we have a strand of DNA before the initiation process begins. The promoter region of the DNA is before the origin of replication, the +1 position on the DNA strand, and it helps guide the RNA polymerase to the correct starting position. Most promoter regions have a consensus sequence, or an average of commonly encountered nucleotides at the -10 region. This is called the Tau box, and there are other promoter regions at the -35 section of the DNA. RNA polymerase is used for all prokaryotic DNA replication because it has multiple parts. The sigma factor on the polymerase is what is attracted to the promoter region. Once the polymerase binds to this promoter region, it begins to unwind the DNA. If there was an insertion of 5 base pairs in between the -10 and -35 promoter region on a piece of DNA, it could cause many problems. The base pairs would alter the location of the -35 region, making it at -40 instead. This would cause problems when the RNA polymerase is trying to bind to the start site. Also, this base pair codes for the amino acid phenylalanine, which can produce problems in the cell if too much of that is produced. The next step of transcription in prokaryotes is elongation. Elongation is when the sigma factor detaches from the RNA polymerase, and therefore the enzyme can move away from the promoter region. The enzyme moves from 3' to 5' end of DNA as it copies the template strand. The strand that is not copied is called the coding strand, and it is the same as the newly made RNA, except RNA has a U instead of T. During elongation, we can also see the transcription bubble and the direction in which transcription moves. In this case, it is from left to right. Lastly, we have termination. We have Rho independent and Rho dependent. Rho independent is when there is a formation of hairpins based on a repeat in base pairs, causing the RNA to bunch up, and therefore the weak U and A bonds in the RNA and DNA causes the RNA to detach. We then also have Rho termination, when the attachment of a Rho moves along the RNA, and when it encounters the RNA polymerase, the polymerase will detach from the DNA, and the DNA will return to normal, and the RNA strand will go off. These are my 2 sources. Thanks for watching.